So for our final one-to-one interview this morning, I'm now joined on stage by Mark Day from Bioasis Technologies. They're a biotech company they've got a proprietary technology platform specifically designed to deliver greater quantities of drugs across the blood-brain barrier and into the brain. Mark, welcome.
Oh, thanks very much, and it's great to actually meet another Brit. So how are you doing?
There's quite a few of us around here. So, so Mark, I want to go to the basics first of all. This might sound like a stupid question, but I'm a journalist, not a scientist. Why is it so difficult to deliver biologics or large molecules across the blood-brain barrier?
Yeah, so I think it's twofold. First of all is the way that pharmaceutical companies tend to develop drugs is a very formulaic, cookie-cutter approach. So very early on, they'll make an assumption based on a particular assay, which is in vitro, as to whether or not the drug can get to the brain. And the number of false positives there are pretty significant. So by the time it gets into phase 1 and 2, you actually find out that you're not achieving your therapeutic dose. And that accounts for about 80% of all of the clinical failures. They've not failed, the drug actually hasn't failed. They've not delivered the right dose. So
Right.
That's like a huge issue.
Is this only related to large molecules, or did we have the same issue? Or are the issues still going on with small molecules?
Yeah, we do see the same issues with small molecules, but not to the same extent as antibodies and enzymes. The issue for the antibodies is that they tend to be large, and you need a way to there's two key components to delivering a medicine into the brain. One is the speed by which, after you inject the drug, it gets to the blood-brain barrier wall. And then the second part after speed is volume. How much therapeutic cargo can you actually deliver? And across the board, when it comes to blood-brain barrier technologies, most of the companies that are out there can only achieve about 0.5 to 1% of the injected dose. Whereas with our platform technology, we're consistently getting 4 to 6% of the injected dose, which results in therapeutic dosages being delivered to the brain. So that's a huge differentiator of our technology.
I was going to ask if there are many technologies out there that are attempting to do this. Obviously yours, but aside for that, we'll come to that in a second.
Sure.
Are there several companies here developing such technologies, or is it quite a small market?
It's a small set of groups, and there's actually a lack of diversity in them. So if you look at, by and large, the most part, work on a particular technology called transferrin, which is an iron-binding protein. Genentech, Roche, Denali, Oceanics, all employ those technologies. And over the last 15 years, they've consistently failed to be able to deliver a medicine across the blood-brain barrier to a patient. And so that's a huge issue, but they tend to be all the same tech. So Genentech originally were the drivers of transferrin, particular technology. Roche, when Roche acquired Genentech, they also became involved with that technology, but independently of Genentech. And then Denali, where Mark Tessier-Levine, who's a great chap, when he left Genentech instead of Denali, he took transferrin with him. So their technology is all the same, so there's a lack of diversity there. But there are 7 or 8 companies that are competitive in the space, but none of them get anywhere near the amounts required, with the exception of ours.
Yeah, it's the XB cubed or XB3?
XB3 or cubed. We call it XB3. And it's a really impressive technology. So the original version of this is a peptide. The original version of it was a 750 amino acid sequence, massive. When I first saw it, I was so big, I was like, there's no way that's getting across the blood-brain barrier. But it turns out that the mechanism of action is through the LRP1 receptor. And that's important because the LRP1 receptor is ubiquitous on the blood-brain barrier wall. So there's loads of doors to sneak cargo in. And then when you get into the brain, when you're actually in the brain, LRP1's actual main function is like cholesterol clearance, so clearing out A-beta in Alzheimer's disease. It's expressed on key parts of the brain in the neurons. And in disease types like with brain cancers, get big upregulation of LRP1. In Parkinson's disease, for example, in the substantia nigra, which is one of the main areas that leads to the Parkinson's symptoms, big upregulation of LRP1 again. So it's a really, really good receptor for getting things across the blood-brain barrier fast and getting them in at a meaningful therapeutic dose.
And was this developed in-house by Oasis, or did you license it or buy it from someone?
Yeah, so the original technology was acquired in 2007 from University of British Columbia, and Wilf Jeffries, who was the lead scientist for this, was the one that made the kind of discovery that, hey, I think this thing can actually move drugs from A to B to C. But that was a huge peptide. My team have developed, they've worked out what the core 12 amino acids were from the big 750 amino acid sequence and got this down to a really, really small peptide that is actually proven to be really important. So the small peptide doesn't interfere with the systemic PK of anything we've bound it to at this point. And on top of that is providing dividends when it comes to the other PK properties. Like, for example, Tmax, we can take a Tmax that was 24 days and turn it into something that in 2 days has achieved its maximum concentration. And actually just this week, MedImmune had just published their independent validation of our technology, which was published in the Journal of Cerebral Blood Flow and Metabolism. And again, we're coming in at the 4 to 6% of the injected dose.